Leptin is an adipokine secreted from adipose tissue and the mucous membrane layer of the stomach. Initial investigations identified leptin receptors, termed OBR, in the brain, which led to the conclusion that leptin is involved in a feedback system related to the regulation of food intake and energy expenditure. Further studies, however, have shown that leptin receptors are expressed in many other tissues, including the placenta, pancreas, stomach, liver, lung, and heart. In these tissues, leptin binding mediates cell proliferation through activation of the STAT3 pathway and the MAP kinase pathway. Leptin is also thought to promote cell survival via activation of the PI3 kinase pathway, upregulation of anti-apoptotic genes such as BCL, and downregulate pro-apoptotic genes such as tumour suppressor gene P53 and BAX. In contrast to adiponectin, leptin levels are elevated in an obese state. This is due to A, an increased number and size of adipocytes secreting the adipokine, and B, the hypoxic conditions created by the expansion of adipose tissue, upregulating leptin expression. Therefore, leptin levels correlate with BMI and percentage of body fat. The first suggestion that leptin might play a pivotal role in tumour development was based on the observation that leptin induces production of cytokines, TNF-alpha and IL-6, by macrophages. Thus, in obese individuals with increased leptin levels, this inflammatory response is upregulated, thereby increasing the risk of obesity-related cancers. Increased circulating levels of leptin have been shown to correlate with cancer progression due to hyperactivation of the growth-promoting and anti-apoptotic pathways. The leptin-induced activation of the STAT3 pathway is also thought to induce angiogenesis of malignant cells via increasing expression of vascular endothelial growth factor, or VEGF. Additionally, leptin is involved in the maintenance of cancer stem cells through regulation of transcription factors NANOG, OCT4, and SOX2. In vitro studies describe that in addition to the elevated circulating levels of leptin, the obesity-induced tumour itself consistently demonstrates an overexpression of the leptin receptor, thereby further exploiting the growth-promoting pathway. Previous epidemiological studies have described that the obesity-induced increase in circulating leptin levels play a potential role in the pathogenesis of endometrial, colorectal, breast, prostate, pancreas, ovary, and lung cancer. A study conducted by Tamakoshi et al. Reported that overweight females in the highest quartile of leptin had a 3-fold increased risk of developing colorectal cancer compared to the normal weight women in the lowest quartile of serum leptin concentration.